So our next speaker will be introduced by Nathan. The next speaker is Sara Walker. Nathan Yee is a mineralogist, biomineralogist, now biogeologist, who is championing astrobiology at Rutgers University, and is one of my colleagues for many years. His background is actually in geochemistry and geology, and he has been becoming, over the last half a dozen, or if not more years, much more attuned to biological processes. And so without further ado, Nathan, it is your turn.
I'm always fascinated to learn about other perspectives of what is life. And we're going to continue exploring this question, what is life, in this section with our next speaker. And it's my pleasure to introduce Dr. Sara Walker. Sara is an associate professor at Arizona State University in the School of Earth and Space Exploration. She obtained her PhD in physics and astronomy from Dartmouth and was a NASA NAI postdoc before joining the faculty at ASU. In her work, she applies theoretical physics to elucidate the informational aspects of living systems. And I'm very excited to hear her talk. Sara, thank you very much for accepting our invitation to speak at the symposium. The stage is all yours.
Yeah, thanks so much for having me. I kind of put a lot of material in this talk because I was so excited about getting to talk with everybody about things, and also because the students in our group are very active and doing a lot of exciting things. So I have kind of 3 parts to the talk, and hopefully I'll get to the 3rd part, but if I don't, I think we'll have enough material for discussion. That'll be a lot of fun. So I wanted to start with my favorite question, which is what is life? Because I think it's really important to kind of articulate sort of the philosophy that one has about approaching this question. And my perspective on it is very much kind of in the territory that we need radically new principles, not even just kind of, applying physics as we know it, but there's actually new physics to learn about underlying the question, what is life? So I am kind of coming around to this view that really what we should be asking is not what is life, but life is what, because it's going to be a very surprising answer. And I think there's kind of a long history of people thinking about the problem from different perspectives. So I have a quote here from Erwin Schrödinger's book, What is Life? About how living matter, while not eluding the laws of physics as established to date, is likely to involve other laws of physics hitherto unknown. And I think in part, maybe what Schrödinger was getting at was the kind of things that Jeremy was talking about in the previous talk. But my interest is actually pushing that a little bit further, because I think that there's some really intriguing features that we see apparent in life that are not necessarily explainable in terms of current physics. And I'm not going to get into a lot of the details of sort of like the deep conceptual foundations and theory behind that. I just wanted to touch on it briefly at the beginning of the talk, because this is a huge motivator for my work and why I do the kind of things that I do. And then I'm going to get a little bit more into sort of technical results on thinking about statistical patterns in chemistry and how we can use that to understand better what life is doing in chemical space. So just to talk briefly about sort of the deeper implications and ideas about the nature of life. Anybody that knows me knows I'm a big proponent of sort of some physics of information underlying life. And I tend to use anthropocentric examples to illustrate why I think that's important. It doesn't mean that I think those are the only examples. I think this is sort of a universal physics. It's just that it's very apparent in what we're doing currently as a technological civilization, and in particular through the acquisition of information that we do as a civilization and doing science. We can control processes that happen in the universe that wouldn't be able to happen otherwise. And so that's really what I mean by this idea of information in the physics of the possible. There are certain things that we observe, like satellites that we launch into space and control that process because we have knowledge of the laws of gravitation, and also that certain elements in the periodic table can be made with high reliability. What I mean is they're not a stochastic one-off event, but there is a reliable process on the surface of our planet, i.e. us with knowledge of nuclear physics, that can produce these high atomic number elements. So if you want to say what kind of physics in the universe can produce these elements and do so repeatedly and potentially in very high abundance, technology is the one that starts to dominate when you get to this very precise control of all the conditions necessary to produce these high atomic number elements. And so the key feature of both of these examples is that you require a long evolutionary process of physical systems acquiring information about their environment. In the case of science, actually compressing those regularities into things that we call laws of physics, and then using those to enable new transformations to be possible in the universe that wouldn't be possible in the absence of that information. And so I would make an argument that these kind of things don't happen by statistical fluctuations, but they require a precise causal chain of events to happen. And so the main distinction between sort of the two domains of physics and why I think this is really important is nicely captured by this quote from David Deutsch, who's at University of Oxford, in a really fabulous book he wrote called The Beginning of Infinity, where he says base metals can be transmuted into gold by stars and by intelligent beings who understand the processes that power stars and by nothing else in the universe. And so I really see this as two domains of physics. One is sort of the physics without us in it, where we study the world objectively and we're not actually a part of the physics and we can write down these sort of regular laws. And then the sort of new physics where we have to understand that there are some physical systems that have knowledge or information and they can harness and control sort of what physical principles the universe obeys. So life doesn't violate the known laws of physics. What it does is actually control them in some way. And if you want to think about this in terms of nanomachines, you might say, well, planets that have, nanomachines on them can do different transformations and different chemical processes that planets that can't, that don't have those. So this is sort of a very broad set of criteria for what life is. It's abstract, but it's necessarily abstract because the idea is to try to get at universal laws that apply to life everywhere, that are universal in the same sense that the laws of gravitation are universal, or the laws of quantum mechanics. And so, so I've been doing a lot of work in this area, which I'll touch on at the end of the talk if I have time to get there, on this idea of assembly theory, and really thinking about how we need to reconceptualize life, not in terms of sort of traditional definitions, but actually, and not tackling defining life, but trying to get at the underlying principles of, laws of our universe that allow us to derive properties that we associate to life. So is there an underlying theory that can explain what life is in this sort of broadest sense? And this picture at the bottom is really supposed to illustrate what that point is that I think that this sort of physics of information that we don't really quite understand yet is a universal physics, but it becomes very apparent in the phenomena we call life. And you might think an analogy, say, to gravitation. We accept that gravity is universal and applies everywhere in our universe, but it's not a dominant force inside an atomic nucleus, but it does control, say, the large scale structure of the universe. And so part of the argument that underlies a lot of the theory development we're doing is that when you get into chemical space and you have a high combinatorial diversity of objects that can exist, there's some new principles at play which actually select out which things do exist. And this is nicely captured by this idea of assembly theory, which is if you take an object like a molecule and you break it apart, there's lots of possible causal histories to construct that object. And that object's complexity in assembly theory is basically associated to the shortest path in that space. So what's the shortest possible history for the universe to produce that object? And the sort of empirically demonstrated results, which I will get to at the end, demonstrates that if we want to actually look at that shortest path in that space, we can make an argument that life is the only thing that can get to sort of high shortest path lengths. And so if you associate assembly index to that shortest path in that space, there's sort of a threshold where the number of steps is so high, you need an object with information about building that object in order for it to even exist in the universe in the first place. So you can think of something like a protein. Even a small protein has a huge combinatorial diversity of possible configurations. If you take a length 200 protein, there's more combinations than there are resources in the universe to make every such protein. So why is it that certain ones exist? That suggests that you actually have a system that knows how to assemble that specific object. And so assembly theory is basically trying to quantify that intuition that things like cups and proteins, oh, sorry, you can't see my cup because of the blur, but there's a cup or a phone or me, don't come into existence in the universe unless you have a specific chain of events. And you can actually measure this in the laboratory in molecules because it's correlated with mass spec. So, so the idea being that there's some threshold where the fact that you have physical systems with information becomes readily apparent as the dominant physics. And so the crux of the argument is that if we actually look at the origin of life transition, there is something that happens above a certain complexity threshold, the assembly threshold in chemical space, where you actually really start to need this sort of new explanatory framework, and this new physics becomes apparent. And most of the work that I've been doing, and this is a lot in collaboration with Lee Cronin's lab at University of Glasgow, is trying to articulate how we can experimentally get at that, which there was a paper that came out in Nature Communications last week demonstrating that assembly theory works as a biosignature framework. But also, more importantly for me personally, is actually trying to get at a deeper explanatory theory for what life is. And a lot of it has to do with this idea that life is basically the physics of objects constructing other objects and the causal chain of those events. And so when we actually see the original life transition, we actually have to start looking in kind of this different reference frame at what the physics actually is. It's not incompatible with the known laws of physics, but it is a really different way of framing them. And just to state sort of briefly, one sort of insight is that really for this theory to work, time has to be fundamental and space has to come out of time. So some of the things with like reversibility and time that come out of thermodynamics and things aren't necessarily, they could be an emergent property of such a theory, but time itself has an arrow and is fundamental. And that's more like causal set theories that are into other areas of physics. So so this transition, I think, is there, and we can actually map it. But what I'm going to talk about mostly in this talk is actually when we go from non-living to living networks, how can we actually tell the difference between a living network and a non-living network and see sort of that this new physics is operating and what would be the signatures of it. And so I'm going to spend most of my time talking about that, and then I'll try at the end to return to some of these ideas about information and assembly and some of the newer work that we're doing with that, trying to map it back to these sort of statistical patterns. So this is just sort of a quick summary of the things that my group works on, mostly for this talk, because I think it's going to be of most interest to this audience. I'm going to be talking about things in the upper left, which is statistically characterizing patterns in life's chemistry at a planetary scale, hence the title of my talk, and what insights that provides for understanding how life is shaped in chemical space. So basically, life, we know, emerges from chemistry. Chemistry is the first scale reality where information actually can become important. Because of this combinatorial diversity, and that information processing structure, if you want to think about those as the nanomachines catalyzing all these chemical processes, actually imprints statistical patterns in the molecules that life uses. And we're looking for those patterns as a way of actually trying to think about what kinds of chemistry for life are possible, because the argument is that the patterns themselves are universal, if not the specific details of the chemistry. So this gets into the topic of universality, which is an important concept both in how we think about astrobiology and thinking about universal biochemistry, but also in physics. So I'm going to touch briefly on how we think about it in those two very different areas and why there's a need for maybe kind of unifying those concepts to really do astrobiology effectively and think about how we're going to start looking for life on other planets. And also at the origins of life. So universality in biochemistry, probably most people are familiar with this concept, is just the idea that all life on Earth, at least that we've encountered, shares component molecules and reactions. So there are some reactions and molecules that are present across all organisms. This is one of the reasons that we can trace the history of life back on in time to the properties of a last universal common ancestor, which shared those basic chemical components. And there's a lot of debate in this community about the nature of the universality that we see. Will all life in the universe share the details, the exact chemical components, which is currently how we conduct our searches for life right now, looking for the components of life as we know it? Or is there some kind of generality about these features that we can actually extract and be able to figure out sort of more universal properties of life? And so this idea brings me to how we think about universality in physics, which is a very different concept. So universality in physics does not care about the component parts. What it cares is about some macro scale feature of the system. And those macro scale features, which are coarse grain features. So it's nice coming after Jeremy because I don't have to explain sort of coarse graining or all these other things. We're well versed in it after his nice introduction. If you think about sort of these macro scale properties, they can be the same and have similar behaviors across very different systems. So a macro scale property is temperature, right? So if you're talking about temperature, it's an average motion of particles in a gas. So it doesn't care about the details of the exact position or momentum of the particles. If we look at it at very different systems, like in a liquid gas critical point, so at the phase transition between liquid and gas, or in a ferromagnetic critical point, we see that temperature has very similar scaling behavior. So this is kind of well known in thermodynamics of phase transitions, that if you look at these macro scale properties in different systems, they might have very similar, or they'll have identical virtually critical exponents as beta. And then that allows us to define a universality class in a physical sense, which is a bunch of systems that behave the same at some critical point. The important feature to make the distinction between how we talk about universality in biochemistry and universality in physics that I want to emphasize here is that universality in biochemistry right now is concerned with the details. What are the specific component parts? Universality in physics is what are these sort of average aggregate coarse grain features that apply across different systems but behave the same. And so what we'd like to do in order to talk about universality in biochemistry, not just for life on Earth, but possibly all life in the universe, is maybe bring in the concept of universality that we have in physics and see if there are ways of applying it to biochemistry. So can we think about coarse-grained or average aggregate features of biochemistry that have very law-like behavior, scaling behavior, potentially, that apply across all living things on Earth and might be candidates for laws that apply to other biochemical systems? In order to be able to approach that, we have to have an ensemble of biochemical systems. And fortunately, because genomic databases have come quite a long ways, it's actually possible to do this. So an ensemble is just, a set of systems that are identically prepared in physics, but we can say at least similarly prepared or representative of the same class of systems in biochemistry. And to get such an ensemble, basically my group has become very adept at mining genomic databases, in particular the Joint Genome Institute for genomic and metagenomic data, and then going through the KEGG database to figure out what reactions are present as being a component of the genetically encoded information in that genome or metagenome, and then constructing biochemical networks based on the reactions that those systems can catalyze. And so the data here, the sort of size of the database is actually not recently updated. I have it here just because some of the work has a smaller amount of data, but it's more recently been updated to include over 11,000 ecosystems and I think 60,000 or so individual species. So these numbers are a lot higher for more recent work we've been doing. And the biosphere, as we identify it here, is just all the cataloged enzymatically catalyzed reactions in the KEGG database. And so that's just a proxy for what the biosphere as a whole is doing in terms of the catalytic potential, what kind of reactions can biochemistry actually catalyze through its enzymes. And so this forms a statistical ensemble that actually has some sense of scale to it because we have individuals, ecosystems in the biosphere, and they are sort of a nested hierarchy of the same system at different scales, but we can also look at the ensemble at each level.
Excuse me, Sarah, I just want to clarify what KEGG means. So,
the Kyoto Encyclopedia of Genes and Genomes.
Thank you.
Sorry.
Yeah, no problem. I just want to make sure that many people that are not familiar with the jargon understand what it is. It's a database which reports what genes are responsible for which biochemical reactions, basically.
That's right. Yeah, so if you have the KEGG database and you have an annotated genome, you can map the functions of the enzymes that are been annotated in the genome to the actual reactions they catalyze. And so this is why we use this pipeline to be able to get these biochemical networks. And there's different ways of doing it, but that's the way our group has been doing it. And happy to discuss with people all those details afterward. And I can also point out, I think Hanju Kim and Pilar Vergelli, who are very involved in this work in our group, are also online, so you can ask them in the chat. I think there might be other people from our group showing up too. So this brings me sort of now to the title of my talk, which is this idea of planetary systems biochemistry. And so sometimes when I have sort of the intro of the what is life from Schrodinger, I always include this quote where he's asking about what's the physics and chemistry, you know, governing what happens inside a cell. And I think actually that's really the wrong way of thinking about what are the fundamental laws that govern life, because life is not what happens in a cell. Life is what happens over evolutionary time scales, and it's not bounded in space and time by a cellular structure. Life is this process of information structuring matter across space and time. And so planetary systems biochemistry is really taking the sort of fundamental unit for thinking about living processes as the planetary scale, which a lot of people have proposed in the past, so that's not a new idea at all. But how do we actually understand sort of the laws that govern biochemistry at this scale? And what we've been focused on so far is mostly looking for scaling laws, because scaling laws have in other areas of biology been demonstrated to be very robust sort of predictors of biological behavior and ways at and allows sort of starting to construct theory to predict why you see certain scaling relations. So a really famous one is metabolic rate scaling with body mass. And they're also, you know, as I was saying in universality in physics, they're one of the ways that we identify universality classes if you have common scaling behavior and common scaling exponents. So what we do with biochemistry is actually look at the size of a biochemical system, which can be different sort of measures, but it might be just the number of compounds that are in the biochemistry that's catalyzed by that organism or ecosystem. Or it might be the number of reactions or the diversity of enzymes. So there's different ways you can think about what the size of the system is from this kind of network theoretics approach. And then we look at some property of the system. It doesn't necessarily need to be a network property associated to the topology. It could just be something as simple as the number of reactions or the number of reactions of a certain type, or a property of the molecule, such as number of chiral centers. So we have a lot of different features that we're looking at. If we look at this ensemble biochemical networks, how do we characterize this high dimensional chemical space and look at what features scale with other features, and can this give us insights into universal principles of biochemistry? So the first sort of work that came out of this set of ideas that our group's been developing, and this was led by Hanju Kim and Harrison Smith, it was to actually demonstrate that biochemical networks themselves, if you look at their topological properties or number of reactions, scale very regularly across individuals and ecosystems as a function of the number of compounds in that biochemical system. So if you look at sort of the entire metabolome, all the biochemistry that an organism or ecosystem catalyzes, you count how many molecules are there, how many different types, and you look at some feature of their reaction, the system level patterns in the reactions, whether that's number of reactions, the shortest distance, which is this L here, between two molecules in terms of number of reactions to get between them, or clustering, which is another topological feature of the network that looks at triangles. You can actually see sort of very regular scaling behavior. So details are in this paper. I'm not going to go into too much detail on this because you can read about it. One of the features that actually we were wondering whether it gives rise to this regular scaling behavior, because we want to know whether these features are characteristic of biochemistry or characteristic of all chemistry, was to try to control for that feature of having universal biochemistry, right? So I mentioned that all life on Earth shares a component set of compounds. They tend to actually be some of in this network representation I've shown in the back here, these high degree nodes are shown large as representative of their degree, how many reactions they participate in. They tend to be things like water and ATP, and those tend to be things that are more universally distributed. So there is a connection between universality and component parts and what the network structure actually is. And this is meant to show that organisms do have this degree of overlap, and each living system is, you could think of it as almost a partition of this global biochemical network with specific frequency of sharing of the component parts across all organisms. So we could say instead, we're not going to share those compounds or reactions in the particular way that we do, and maybe we'll just randomly sample the space of chemistry that biochemistry uses. So this is one way of constructing a random chemical ensemble, but it's one that is constrained to being in the same region of all chemical space as biochemistry as we know it. And the reason for doing that was it's very easy to construct such an ensemble. Constructing really random ensembles is very hard because you have to go to chemical databases, and there's lots of issues with that, but I'll talk about that a little bit later. In any case, what we did to try to understand whether these scaling laws were unique to biochemistry or a generic property of chemical space more generally was randomly sample reactions and generate scaling behavior for networks that were generated from this random sampling. And so the original data is shown in gray, and the red or the orange data is actually the randomly sampled reactions. And what we find is there's still scaling behavior, but it's statistically distinguishable from what we see in life. And in many cases, you can definitively tell the difference for any particular measure between a real biochemical network and these randomly constructed ones. And in fact, by these kind of scaling relations, you can actually tell, and this sort of the average topological properties, you can statistically tell the difference between the domains. So there is some variation even within life, but life also has a sort of universal property to these networks. If instead we do random sampling where we have a frequency-based selection, so we're actually trying to choose reactions based on how commonly they occur across known life, then we recover more of the scaling features that we actually observe in the real biochemical systems. But in some cases, these are still statistically distinguishable, so it doesn't recover all of the features, but the majority of the features. And so just to summarize that, we have this sort of scaling behavior that we see for life on Earth. It seems to be predominantly controlled by the fact that life on Earth shares a subset of reactions. We see that sort of reinforced when we do genome level sampling that recover most of the features of ecosystems. So the main take home for this is sort of, we might call it a conjectured principle of planetary systems biochemistry, is that the scale invariance of biochemical networks that we see, the particular features of that scale invariance are actually require a universal set of biochemical reactions. A subset of the reactions have to be universally shared across all life in order for ecosystems to have the same topological structure as individual organisms. And for those familiar with network theory, that might not be too surprising, because if you merge scale-free networks together, which biochemical networks are not exactly scale-free, but they're almost scale-free. If you merge them, it's not guaranteed that you're going to necessarily get random networks when you keep adding them to, or scale-free networks when you keep adding them together. You might get random networks instead. So there are some features of this overlap between reactions and biochemistry that allows biochemistry to have the same features across different scales. Okay, so we see regular scaling behavior, and when physicists see scaling behavior, they think phase transitions and criticality, or think about different kind of, states of systems having different scaling relations. So, so one of the things that our group started thinking about is if these networks are really controlled by this subset of universal biochemistry, is there some way of thinking from this network theoretic perspective about what the last universal common ancestor was as a phase transition in the network properties of the chemistry that life uses? This idea also itself is not necessarily new. Nigel Goldenfeld and Carl Woese have been thinking for a long time about this idea that there was some kind of phase transition in early life associated with the collective behavior of populations of cells that led to what we call the last universal common ancestor and the bifurcation into the three domains of life. And a lot of our work in our group is actually very suggestive of the kind of properties associated with that phase transition. I think one of the things that's maybe a little bit different is they were thinking about LUCA as an ecosystem scale property, but a lot of the features that we're looking at, because we're doing this sort of planetary scale survey, suggest that we might even think about it as a planetary scale phase transition and sort of the chemistry that was catalyzed by life and the particular structural properties associated with the networks and the kind of reactions used and their statistics. The last universal common ancestor exists today. It's not a single organism anymore. It's distributed over the entire planet. It may never have been a single organism. Like I was just arguing, it probably was distributed over the entire planet to begin with. But it's sort of a problem for original life studies that if we want to genomically reconstruct life, early life, we're limited by how far back in time we can go genomically. So the last universal common ancestor is kind of the earliest piece of history we have if we are reconstructing from modern life backwards in time because of this genomic boundary. So are there ways that we can actually use the properties of biochemistry distributed across the planet and these kind of ideas of looking at these statistical patterns or universality to actually infer what happened earlier than the genomic record allows us to? So this is a question I'm really interested in. It's in part inspired by the idea of the last universal common ancestor as like a surface of last scattering. So it's like the information is here, it's universally distributed. I just want to make a quick point before I get to that though, which is that in modern systems, LUCA is actually an ecosystem scale property. So I have LUCA data on here. What I mean by LUCA, the LUCA model that we have and have been using is a consensus model that was constructed by Aaron Goldman based on sort of the 8 leading models of the last universal common ancestor. And so we have details of it in a preprint that I'll cite in a few minutes. That's up on the bioRxiv and currently under review. So I can refer you to that paper if you're interested in the details of sort of what those models are and how the consensus was constructed. But we use that model basically as the model of LUCA. And we can look at sort of across all of our modern genomes and metagenomes, where are most of the LUCA reaction, like where is most of the LUCA biochemistry located? And I'm going to show a few plots that look like this, where we plot universality versus rank. And basically what we do is we rank order enzymes, reactions, or compounds based on how frequently they occur across a data set. So if a enzyme, as shown in this plot, occurs across 100% of our data, then it gets a universality score of 1. So the flatter this curve is toward 1 on the top here, or well, these are rank ordered by the highest, it gets a universality score of 1 here. So the flatter this curve is, the more universally distributed that data set is. So what this is saying is that enzymes are more universally present across ecosystems than bacteria, archaea, or eukaryotes. But it allows you to actually quantify those differences. So that might seem intuitively obvious. You find more enzymes in common across ecosystems than you do across individual organisms, but it allows us to look at that a little bit more rigorously and actually look at the patterns. And what we see is, you know, also maybe intuitively obvious, but nice to see the statistics, that the LUCA biochemistry is more universally present in ecosystems than individuals. And then, as I was just saying, at the genomic level, LUCA is sort of like a surface of last scattering. So this is a picture of the cosmic microwave background. The reason I'm using that is because that's sort of the last history of the surface of the early universe, where we can actually get information directly from photons, because before that, the universe was not transparent to photons. And it's a little bit an analogy to genomic information. There's a certain surface, we call that the last universal common ancestor across life on Earth, across the surface of our planet, that we can't infer based on that method directly. But if we identify sort of the rules of the chemistry and how life behaves, we might be able to infer what happened earlier than that, in the same way that we know what happened in the first few minutes of the universe, at least to fairly high precision, based on knowing the laws of physics. So we need to know the laws of biochemistry to get further. So this brings us back to the idea of universality in physics. And maybe if we can identify these scaling laws, we can actually see if the last universal common ancestor, as we've constructed it so far based on phylogenetic reconstruction, is consistent with those universal patterns that we see in the biochemistry. And if not, are there ways that we can use these scaling laws to construct new LUCA models and basically build an ensemble of possible biochemistries that are consistent with our planetary history and what was happening at that time. I don't need to explain what coarse graining is again, because Jeremy did such a nice job. So I'm going to kind of skip over this, but universality usually comes from coarse graining. It's a macro scale property when we're talking about it in physics. And so what we need to do is coarse grain biochemistry. So one way of doing it is based on the network properties that I was showing before. But another way to do it is just by looking at enzyme functions. And so I don't want to get into details of this because I don't have enough time, but if we look at the first digit of the classification of enzymes, there's actually 6, there's now 7 in sort of these enzyme databases, classes that are sort of the major categories of enzyme function. And we use that as a coarse graining of biochemical space to try to see if there are regular scaling behaviors associated with biochemistry and the functions that reactions have. So these are the 6 major classes. Some of the genomic information doesn't necessarily have an EC associated to it, so we don't use that in our data pipeline. And then basically what we do is we look at all the genomes we have, we count how many of each EC are encoded in the genome or metagenome, and then we look at how many of those enzyme functions there are versus the total number of enzyme functions in that biochemical system. And we see very regular scaling behavior for the enzyme classes. So you can associate law-like scaling behavior to the use of these macro scale coarse-grained functions that biochemistry uses. Some of the enzyme classes are super linear in their scaling behavior. Oxidoreductase, as you might expect. So what this basically is suggesting is that larger biochemical systems need more redox functions than smaller ones. And some of them are sublinear. So for example, ligases, as you increase the size of the biochemical system, the proportion of ligase functionality decreases sublinearly. And so these define actually universality classes. Some of the enzyme functions are superlinear across all of our data sets. Some are sublinear across all of the data sets. And we can think about these as candidate universality classes for biochemistry. Now, one of the questions you might have is, can we actually say these are universality classes in the physics sense, where they're independent of the specific component details? And so to test this, we went back to that idea of these universality plots that I had shown before. And so this is just showing for each enzyme class how universally distributed are functions in that class across our different data sets. So you can see it's pretty obvious that ligases are commonly shared across a lot of the data sets. So they have a high universality score for a lot of enzymes. Functions, and oxidoreductases are less universally distributed. So that's sort of the only thing you need to get out of this slide. And what I want to show is sort of the important point here is we can take the scaling coefficients that we have here, and then a score we associate to the area under the curve for the universality, which is a measure of how universality the component membership is. So this is universality in physics. This is universality in biochemistry. And we can show that they're not correlated. Which means that the universality that we see from these scaling laws is actually an emergent property of the networks as a whole. It's not tied to the specific component reactions and how commonly they occur across life. So this is very suggestive that these are actually macro scale constraints on the organization of biochemical systems at the level of what the ratio of different functions are that they use, and not a feature of the fact that there are common reactions used across life. We can also ask questions now, like I was suggesting, about the last universal common ancestor. And so current LUCA models in the same universality class are not as modern life. Effectively, they are almost in the same universality class. So if we take those scaling laws that we had for each of the domains and we extrapolate down to the size of the last universal common ancestors, biochemistry, number of ECs that we have in these LUCA models, some of the scaling behaviors predict what we would expect. So for oxidoreductases, for example, it might be, the right number for that size biochemical system, but LUCA is heavily overrepresented in ligase functions, which is probably expected because ligases are more universally shared. So when you do a phylogenetic reconstruction, you're biased to include more ligases because they tend to be more universally shared components, therefore inferred as having an earlier history. So, so what this suggests is that there is some agreement between what we see in LUCA and what we see with the scaling laws, but these scaling laws might allow us to infer sort of or constrain properties of the last universal common ancestor in its biochemistry, and potentially construct sort of different likelihood distributions for the models that might be appropriate as the biochemistry of the last universal common ancestor, or at least that's one direction we'd like to go. So a second conjectured principle of planetary system of biochemistry is that biospheres exhibit universality and reaction functions that are independent of the details of the component chemistry. And that's supported basically by this lack of correlation between the scaling laws and the frequency of the components that biochemistry is using. I'm going to stop for a second. How much time do I actually have left? Because I want to have time for discussion. I have too much stuff in my talk as usual. So I can stop here. It might be a natural stopping point depending on how we are for time.
So I believe you have another, there's going to be one more talk after you, but this would be a good time to maybe take a breather, because there are questions in the
I'm going to go to my thanks slide. So hang on a second. We'll just, I just want to, yeah, that sounds great. I'm happy to take questions here. I kind of.
So Jana, I saw you had a technical question. I'm just going to call you out, and maybe you can ask your question in person. Jana, are you there? Okay, so I will read the question out. So there was a question about your network structure properties. So the question is, how many of the observations of network structure properties is driven by incomplete annotations and scientific focus on the known functionality?
Yeah, so we've thought about that a lot. I think the best way that we can get at it with our data set is to basically remove reactions, because we don't know what missing function there is, but we can remove functions that are there. And when you do those kind of perturbations where you remove a certain percentage of your data set, the patterns seem to be pretty statistically robust. And I think part of the reason that we're focusing on these network properties and statistical patterns, and in particular, like sort of high level patterns in the enzyme functions, is that they tend to be more robust to missing data than if you were looking at specific details of the chemistry. And in fact, what I'd like to be able to do with it is to use these patterns to predict what are the missing functions that haven't been annotated in a particular data set. So far, the best we can do is look at missing, removing functions from the data. I'd like to, we've talked in the past about looking at better annotated data from your databases, and I still want to compare, sort of these less well annotated systems to the better annotated systems. So that's another path that we still have to do.
So Sarah, I'm going to invite you to type your responses to the chat. I'm very selfish because I want to ask my own questions in the last 2 minutes. Okay. So I'm fascinated by this subject, and I just want to hear your thoughts on how these new laws of life that you've discovered can inform NASA missions. So these are missions to search for life in the universe. So what are your thoughts on applying these new findings to a real life astrobiology investigation?
Yeah, there's two features I think I'd like to focus on. One of them has been experimentally demonstrated, which I didn't exactly talk about, but I'll just show that slide real quick since you asked the question. Sorry, too many things here. So this assembly measure that I mentioned is in this paper, which came out in Nature Communications last week for people that are interested. And the green data set here is actually sort of the distribution of molecular assembly that we see for biological samples, and the other samples are dead or abiotic. So what the paper is basically suggesting is if that we apply this assembly theory to complex molecules, we can identify which ones are unambiguously produced by an evolutionary process, meaning that they have too many steps in their formation to actually occur spontaneously in any environment. So that's one feature that is ready to fly because it's basically based on using mass spec to actually infer assembly. The other one that I'm interested in doing is actually trying to use these scaling constraints to basically use like combined with machine learning and cheminformatics to basically grow biochemical networks on other planets. So take planetary context and the constraints of the environment to kind of predict what are the possible biochemistries on a particular planet, if they exist, and then use those sort of as another backdrop for biosignature detection, because you would know sort of the signature in that environment if you had a system with these kind of constraints operating on it and what kind of molecules would be there. And I think that combined with the molecular assembly would probably be, you get these system level properties, but also properties in the individual molecules. That's the short answer.
So Yori, I see you with your hand up. Do we have time for a quick question? Please go ahead.
Yeah, there's only one LUCA. Is that by chance, or there's a chance to be more than one LUCAs?
My personal perspective, which is very informed by the work that our group has been doing, is that there's one LUCA per planet. So you have to have a consensus biochemistry for a planet in order to enter like what we would call an open-ended evolutionary process. So, so I think about planetary scale biochemistry really as an integrated system, and individuals are instances of that in time and a particular space, but you have to look at the entire system and how it's interacting. And that's really what our work is capturing, is the features of that system and those interactions at this very coarse grain scale.
So I'm sorry, I'm going to steal 5 more minutes from our next speaker. Our next speaker is going to give a very short presentation. So I've spent a lot of time thinking about this question myself, which is, were there multiple origins of life? And it's very interesting that if there were, one form of life came to dominate the planet, so much so that even if there were other origins, it caused their complete extinction. So we no longer see other forms of life. So what are your thoughts on that? If there were multiple origins, is this one form of informational translation in terms of DNA, RNA, protein? Is that, what are your thoughts on that? Multiple origins versus a single origin?
I think I would think about it slightly differently, which I think a lot of people think of like life happening in chemistry and then increasing the scale of what life is by like, you know, through evolutionarily, you know, like sort of creating a diversity of species and then ecosystems and then these planetary scale phenomena. The way I think about origins of life is a multi-scale phenomena. So it's a process that's happening across a lot of scales at once. And you actually need the interaction of scales to drive what we call the origins of life. And so it can kind of look like multiple origins, but once you start, when you increase the scale of the system, it's one origin. So there's an origin of one biosphere per planet, but the sort of local events that were happening were probably you know, a multiplicity of events, and the constraints between those scales are actually what led to sort of the biochemistry that we have now. If that makes sense. So I think it's sort of a reframing of it as, you know, this process that happens at a single scale, and we have to look at it radiating out to thinking about this process that's happening simultaneously across different scales. And the last universal common ancestor is really this, in my mind, is this planetary scale phase transition when the networks are like, and their interactions actually acquire certain properties to make that system robust over evolutionary timescales. And so if you think in like the artificial life literature and the way people talk about, you know, principles of life there, that's really the transition where you have open-ended evolution. So you might have a lot of failed biospheres that never make that planetary scale transition. And they wouldn't be life like we are. They'd be very localized life, which to me is much less alive than the kind of planetary phenomena we have.
This is great. So what I'm going to do is, thank you very much, Sara. If you're generous enough, please continue this discussion on the chat. I am going to introduce our next speaker, after which I would like to invite Jeremy and yourself and Aaron to maybe have an additional few minutes of discussion. But I would like to move on. Thank you.